Okay, this is Professor Kurinchi Guruswamy, and I am talking about the clinical relevance assessment of animal preclinical research, or AA tool. This tool was developed by the team that is shown in this slide. And what are the objectives of this particular presentation? It is to provide a bit of background about the tool itself and how it was developed, and then subsequent presentations will look at the explanation of the RAA tool. And the purpose of this particular exercise is to obviously provide you knowledge, and then you can pilot this tool. And once you pilot this tool, we will measure the inter-rater reliability assessment and also how long it takes for someone to complete this tool. Okay, little bit of background and development. Only a small proportion of animal preclinical research translates into human benefits. Of 101 promising technologies that includes drugs, devices, and gene therapy, only 27 were subsequently tested by human randomized controlled trials. And only one technology provided clinical benefit. In another study, of 100 potential drugs giving objective improvement when evaluated in a commonly used mouse model for the treatment of amyotrophic lateral sclerosis, none were found to be clinically beneficial. In another study, a systematic review found that there were significant differences in the estimates of treatment effectiveness in animal experiments compared to that observed in RCTs, with some interventions being beneficial in animals but harmful in humans. Why did all of these happen? It could possibly be related to the design, conduct, and reporting of preclinical studies. So, what did we do? We performed a literature search. We searched the Equator Network's library of reporting guidelines for guidelines that describe the design, conduct, and reporting of the preclinical research. We searched the references of these guidelines to identify further relevant publications. We identified 21 publications, and the main topics covered included bias, random errors, reproducibility, reporting, or a mixture of the above. Now, for people who are not familiar with what these are, don't worry, all of this is explained in later presentations. Before we go on to how the tool was developed, we need to explain what a domain-based tool is. Domain-based tool is a tool that assesses different aspects. These different aspects are called domains. So the tool assesses different aspects that impact the outcome of interest. And in this particular situation, the outcome of interest is clinical relevance of preclinical research. Now, such domain-based tools are preferred by methodologists to assess clinical studies. No domain-based tool for assessment of clinical relevance of preclinical animal research exists. So after having established that there is no domain-based tool for preclinical research, we recorded the topics covered in the previous guidance on preclinical research to develop a list of domains and signaling questions to be included in the formal domain-based assessment of preclinical research. We included signaling questions for each domains. And what are signaling questions? Signaling questions are questions that help in the assessment about a domain. Therefore, it is the domain that's important, or the domain assessment is important, rather than the answers for individual signaling questions. Now, depending upon the nature and purpose for the research, certain domains may be more important than others. For example, if the purpose is to find out whether there is enough information to perform a first-in-human study, the clinical translatability and reproducibility domain is of greater importance than if the report was about the first interventional study on a newly developed model. Okay, so don't worry about the domain names. We will be explaining these in subsequent presentations. We then approached experts. These are preclinical and clinical research experts, and we sampled them using snowballing principles. It is of note that all had interest in improving the clinical relevance of preclinical research. Then we conducted a modified Delphi consensus method to refine and develop the tool. We conducted 3 rounds, and the score for each domain and question ranged from 1 to 9, 1 to 3 indicating least important question or domain, and 7 to 9 indicating most important question or domain. If greater than 70% thought that a domain or signaling question was important, they were included. Then we refined these signaling questions and explanation by iterative electronic communications. So what do we require from you? We want to pilot the developed tool with regards to whether different people arrive at the same conclusions or not. Now the plan is to provide explanation for domains and signaling questions, and that would be based on this presentation, this PowerPoint presentation, and which has been split into multiple videos. And we'll also demonstrate the clinical relevance assessment in one preclinical research study, and the link is provided at the end of this presentation. And then we want you to assess a preclinical research study. Now, full text is available for 2 studies by clicking on the link again available in this presentation. Please choose one of these, anyone will do. We'll collect this information from several people. We will assess the inter-rater reliability based on the answers for domains, but also assess this for each signaling question. You can then contact the team for clarifications related to the tool. You can also contact the team for improvements in the explanation provided for the tool. Please record the time taken for completing the assessment of the study that you have chosen and provide this information to us. Okay, what will you get from this? Your knowledge on the factors that improve your study's clinical relevance will be more after completing this tool or after completing the assessment of a study. You will be part of a development of a tool on clinical relevance of preclinical animal research, and you will be acknowledged in any publication that arises from this. You can also potentially involve in projects that answer some of the uncertainties mentioned in the explanation. And if you do have interest, please express your interest in these uncertainties. You can also potentially involve in similar projects of other fields of preclinical or clinical research. And again, please express your interest in the fields that you want to be involved. Now a little bit of background about the tool itself. Now what's the scope of the tool? It is to be highlighted that this tool is only for assessment of the clinical relevance of a preclinical research study in terms of the likelihood that therapeutic preclinical findings can be translated into improvement in the management of human diseases. This is not a tool for assessment of the quality of the study. For example, how well the study was conducted. Why is it important to make this distinction? Even a very well designed and conducted preclinical study may not translate to improvement in the management of human diseases. Of course, this is the case in clinical research as well, where very well designed and conducted clinical studies may not result in improvement in human diseases. Okay. This tool is also applicable only in vivo laboratory-based preclinical animal research evaluating interventions. It is not intended for use on other forms of preclinical research, for example, in vitro works, example cell cultures, in silico work, or veterinary research. And it is not applicable in the initial exploratory phase of development of new animal models of disease, although the tool is applicable in interventional studies using such newly developed models. Okay, so to summarize, this tool can be used only for preclinical animal research, and it is not applicable in the initial exploratory phase. The tool itself has 8 domains and 28 signaling questions. And how do you classify the signaling question? Each signaling question, can be classified as yes, probably no, no, or no information. With this regard, yes and probably yes equates to the same results later on in the domain classification. No and probably no are also equivalent in terms of how the domains will be classified. There are a few questions that can be classified as not applicable, and these questions usually start with the phrase if. How do you classify the domain? You classify the domain as low concern if all the signaling questions under the domain were classified as yes or probably yes. High concern if any of the signaling questions under the domain were classified as no or probably no, and moderate concern for all other combinations. If you are using the Excel file that I have provided, this is done automatically. In other words, if you put in the signaling question classification, the domain classification is done automatically. Although you can manually override the classification if you feel that's appropriate and provide some explanation as to why you did that. How will you classify the overall study? If it is low concerns for all the domains, then the study has high clinical relevance. Otherwise, the study has uncertain clinical relevance. As mentioned before, not all domains are important in all types of studies, and the relative importance of the different domains may depend upon the purpose of the study. The overall study classification is also done automatically if you are using the Excel file. However, you can also manually override the classification. This is the first look at the tool itself. You can see that there are domains and there are signaling questions under each domain. All of these will be covered in subsequent videos as to what they mean and so on. So I'm not going to go through the entire list now. So this slide shows domain 1 and 2, and this slide shows domains 3 to 6, and the seventh and eighth domain are shown in this slide. Now, how to use the explanation that's provided in this presentation. Now, descriptions are provided before the questions if necessary. And in terms of classification of domains, I've already mentioned yes or probably yes are equivalent, and same way, no or probably no are equivalent too. When some information is not reported or not clear, the classification is no information. But when it is clear that there is a concern about the issue, then it's no or probably no. When it is clear that there is no concern or the concern about the issue is low, then it's yes or probably yes. Now, while classification, the person assessing the clinical relevance, that's you, the assessor, is not expected to search for information in addition to what is provided in the text or supplementary information. For example, if the authors say something that this test is accurate, then you're not expected to search for studies that demonstrate the accuracy.